The last video dealt with the very earliest steps before life on Earth, turning inorganic molecules into organic molecules. This video outlines how the next steps might have happened. It's video 3.4 on the origin of cells and endosymbiosis. And it's all about these two objectives. Pause to see more. So the origin of life didn't happen in just one step, and it wasn't a quick process. Earth was around for about 700 million years before there was life. This video will connect spontaneous abiotic origin theory to living cells. The Miller-Urey experiment showed how life's building blocks, organic monomers, could have arose in early Earth conditions. Next, those monomers had to combine into polymers. The polymers had to assemble into cells, and then those cells had to go from simple to more complex by evolution. In their experiment, Miller and Urey found amino acids. Amino acid monomers are the building blocks for protein polymers. The other macromolecules essential for life are lipids, carbohydrates, and nucleotides. If amino acids could have formed under early Earth conditions, then why not the other macromolecules? Now, nucleotides are the building blocks for DNA and RNA, and there's growing evidence that RNA was the first genetic material and not DNA. And if you think about it, the origination of genetic material was huge. Genetic material makes inheriting traits possible, which makes evolution possible. Instead of each species having to restart after each generation, RNA allowed for successful traits to be saved and act as kind of a starting point for the next generation. Next to those macromolecule polymers were thought to have self-assembled into primitive cells called protobionts. Protobionts are basically a membrane with protein inside, and having a membrane bubble allowed processes to happen on the inside of the bubble that were different than the processes outside. Now, later, those protobionts are thought to have evolved into the first, simplest prokaryotic cells. Finally, prokaryotic cells gained complexity to make them eukaryotic cells. This is going to be the main focus of this video, so I'll save the details until later. The rest of this video is dedicated to the origin and evolution of cells. One hypothesis about the origins of cells is that the ocean became a soup of macromolecules, a mixture of all of those molecules essential for life. Because of biochemical properties we'll talk about in a couple units, lipids, or fats, like to be next to each other, especially in water. Keep in mind that lipids are the main components of membranes. Scientists think that lipids self-assembled into these membrane bubbles. The bubbles contained other biopolymers like RNA, proteins, lipids, and carbohydrates. And once the bubbles started to function differently because of what was inside, we call them protobionts. Protobionts are the precursors for cells. There are a couple of important things going on. First, the membrane of protobionts separated the inside from the outside. And why is this important? Well, the inside becomes an environment favorable for metabolic chemical reactions, even if the outside is not favorable for those reactions. Second, protobionts can have their own genetic material. Then, when protobionts self-replicate, another huge step, protobionts pass genetic material to the next generation, essentially saving all the favorable traits and creating a pathway for evolution. Now, scientists have done proof-of-principle experiments with protobionts. With a biopolymer mixture in a test tube, scientists found that membranes do spontaneously form these bubbles. And not only that, but the lipid bubbles did metabolic chemical reactions and even reproduced. Now, does this prove that cells came about this way? No, but the experiment shows that the idea is feasible. If we accept that protobionts were the early cells, then protobionts were the starting material for the most simple cells, prokaryotes. And this is where having genetic material is huge. Mutations in genetic material could make protobionts complex until eventually cells ended up as prokaryotes. All right, now I've been using prokaryote like you should know the meaning, but I want to make sure. The two major cell types are prokaryotes and eukaryotes, and here's a whole list comparing the two. To simplify, think of prokaryotes as simple cells with no nucleus and eukaryotes as more complex cells with a nucleus. Prokaryotes are simple because they don't have membrane-bound organelles, and eukaryotes do have membrane-bound organelles like endoplasmic reticulum, Golgi apparatus, vesicles, vacuoles, and more. Finally, it's a good idea to have an example of each. A good example of prokaryotes is bacteria, but it's cooler to say a specific bacteria like E. coli. Eukaryotic cell examples include plant cells, animal cells, yeast, and protozoa, and fancier examples are like neurons, sperm cells, egg cells, amoebas, and paramecia. So, how did nature bridge the gap between prokaryotes and eukaryotes? Well, one idea is called the infolding theory. Infolding theory explains the origin of membrane-bound organelles. And the idea is that the outer membrane of prokaryotes folded in and broke off to form inner compartments. Those inner compartments were the early versions of organelles, things like the nucleus and endoplasmic reticulum. A good thing about organelles is that each organelle creates a specialized compartment with a microenvironment specialized for a certain cell job. Since it was advantageous to have these infolded structures, cells with infolding stuck around as modern-day eukaryotes. Yet another theory explains how eukaryotes got chloroplasts and mitochondria. Chloroplasts and mitochondria arose by a different process called endosymbiosis, and endosymbiotic theory outlines the origin story of mitochondria and chloroplasts. Endosymbiotic theory suggests that early eukaryotic cells engulfed specialized bacteria. These bacteria gained the advantage of being protected from the environment by living inside of a eukaryotic cell, and eukaryotic cells benefited by bacteria helping out with a specialized job. Aerobic bacteria are specialized at making ATP. Aerobic bacteria make ATP 18 times better than anaerobic bacteria. Can you imagine being able to do something 18 times better? Well, it's thought that these aerobic bacteria eventually became mitochondria, the specialized ATP-making organelle of cells. Photosynthetic bacteria are specialized to make food, glucose, from 3 common ingredients: carbon dioxide, water, and light. Now, for a cell that doesn't do photosynthesis, engulfing a photosynthetic bacteria is like having a chef live in your house. It's thought that these photosynthetic bacteria eventually became chloroplasts, the photosynthetic masters of eukaryotic cells. Let's check out these ideas more closely. Here's the origin story of mitochondria. At first, there's a simple eukaryotic cell. That early eukaryote finds an aerobic bacteria cell and engulfs the cell by a process called phagocytosis. Normally, phagocytosis is how single-celled organisms get their food, but this cell didn't digest the aerobic bacteria. Instead, the aerobic bacteria lived inside of this tiny bubble in the eukaryote. The bacteria gained protection from environmental hazards, and the eukaryote gained the ability to make ATP 18 times more efficiently. And here's how chloroplasts arose. This eukaryotic cell has mitochondria. Now, that cell is engulfing a photosynthetic bacteria cell, but not digesting the engulfed cell. Again, the engulfed bacteria benefits by being shielded from environmental hazards, and the eukaryote benefits by being able to make its own food by photosynthesis. In other words, the engulfed bacteria was preserved as a chloroplast. Eventually, the relationship between the eukaryote and engulfed bacteria evolved so that they couldn't be separated from one another, and at that point, we'd consider the engulfed bacteria mitochondria and chloroplasts. A fancy name for this relationship is obligate symbionts. What this means is that at some point, if you took a mitochondria or chloroplast out of a eukaryote, neither would be able to live on its own like the original bacteria would have. Endosymbiosis is a cool theory for many reasons. First, endosymbiosis is a cool fusion of all of this undeniable evidence. Second, endosymbiosis theory was championed by a female scientist with Chicago connections. When you think about the evidence for endosymbiosis, it might be easier to categorize the evidence into structural evidence, genetic evidence, and functional evidence. Let's unpack each one in more detail. Now, this is what I expect you to be able to do with the evidence for endosymbiotic theory. First, describe each specific piece of evidence, and second, explain how that evidence supports and connects to the endosymbiotic theory. First, structural evidence. Basically, mitochondria and chloroplasts look like bacteria. And what do I mean by that? Well, mitochondria and chloroplasts, abbreviated as M and C, have a similar size and shape as bacteria. Now, that's not strong evidence by itself, but there's more. Mitochondria and chloroplasts have internal structures that independently living bacteria have. These structures include their own DNA and their own ribosomes, and what's more is that the structure of the DNA in mitochondria and chloroplasts is like bacterial DNA. The DNA of mitochondria and chloroplasts is in a single circular chromosome, just like in bacteria. In contrast, the DNA in eukaryotic cells' nuclei is in several linear chromosomes. So that's the describe part of the evidence. But what about the explain part? How does this support endosymbiotic theory? Well, similar structures are indicators of evolutionary relatedness. Since mitochondria and chloroplasts have similar size, shape, ribosomes, and uniquely shaped DNA as bacteria, that suggests that mitochondria and chloroplasts share evolutionary lineage with bacteria. The presence of DNA in mitochondria and chloroplasts is genetic evidence as well. Remember that eukaryotic cells have their own separate DNA. Mitochondria and chloroplasts have their own separate DNA. And DNA in mitochondria has genes that encode proteins for aerobic cellular respiration, and DNA in chloroplasts have genes needed for photosynthesis. Now, how does that support endosymbiotic theory? Well, having DNA is a characteristic of all living things. So having separate DNA suggests that mitochondria and chloroplasts were once separate living things, like bacteria. Another weird thing is that mitochondria and chloroplasts reproduce independently from the cell. Now, eukaryotic cells reproduce by a process called mitosis, but mitochondria and chloroplasts can reproduce even when mitosis is not happening. And that reproduction process looks very different than mitosis. And in fact, mitochondria and chloroplasts reproduce by a process that looks a lot like binary fission, which is how bacteria reproduce. How does this support endosymbiotic theory? Well, the fact that mitochondria and chloroplasts reproduce on their own, a characteristic of living things, and that this process looks like bacteria reproduction, suggests that chloroplasts and mitochondria are related to and were derived from separate living bacteria. So we've completed this final link for the story of this video. These theories of infolding and endosymbiosis account for the transition from simple prokaryotic cells to complex eukaryotic cells. So let's recap. The story of life's beginnings was a multi-step process that took up to 700 million years. Once organic molecules assembled into polymers, then the polymers had to assemble into primitive cells called protobionts. Genetic material and reproduction allowed protobiont evolution into prokaryotic cells. Then, prokaryotes evolved into more complex eukaryotic cells, first through infolding to create most organelles, then through endosymbiosis of bacteria to generate chloroplasts and mitochondria.